Biomolecules. All living organisms are made of the same chemicals, elements, and compounds. Biomolecules are all the carbon compounds that present in living tissue. That present in living tissue. These molecules are involved in the maintenance and the metabolic process of living organisms. Cellular pool is the sum total of different types of biomolecules, compounds, and ions present in a cell. The elemental analysis of the plant tissue, animal tissue, or a microbial paste shows the list of elements like carbon, hydrogen, oxygen, and the several others along with their respective content per unit mass of a living tissue. The list of elements present in the earth crust is similar to the elements present in a living tissue, but the relative abundance of the carbon and hydrogen compared to the other elements in living organisms is higher than the earth's crust. How to analyze the chemical composition? For chemical analysis of the organic compounds, a living tissue is taken. Is taken and churned with the trichloroacetic acid, which is to obtain a thick slurry. Two fractions filtered acid soluble and the retentate or the acid insoluble pool are obtained on straining with the cheesecloth. Acid soluble pool contains thousands of the organic compounds. These are further isolated and purified. Through analytical techniques, molecular formula and structure of compounds can be detected. Inorganic compounds can be analyzed by burning a living tissue after it is dried out. After organic compounds are burned and released, the ash that remains consists of the inorganic metal elements like the calcium and magnesium. Inorganic tissues can be analyzed by burning a living tissue. After that, we can obtain elements like calcium, magnesium. Therefore, elemental analysis gives elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compounds gives an idea of the kind of organic and the inorganic constituents present in the living tissues. Inorganic constituents of the living tissues are the potassium, calcium, magnesium, water, and compounds like NaCl, calcium carbonate, sulfate, potassium, phosphorus, phosphorate, I guess. Before. Amino acid. Amino acids are the organic compounds that contain an amino group and an acidic group as substituents on the same carbon. An amino group and an acidic as substituents on the same carbon. amino group and an acidic group. That is the alpha carbon. Therefore, they are called the alpha amino acids. They are substituted methanes. There are four substituent groups occupying the four valency positions. These are hydrogen carboxyl group, amino group, and a variable group designated as R group. The R group in amino acids could be a hydrogen such as glycine, a methyl group such as alanine, hydroxymethyl such as serine. Oh my, darling, my vitamin. There are four substituent groups occupying the four valency positions. Substance on the same carbon. That is alpha carbon. That way they are also known as alpha amino acids. Okay, these are there are four substance group occupying the four valency position. Mono group and the variable group designated as R group. The R group in amino acids could be hydrogen such as glycine, or methyl group such as alanine. R group can could be a hydrogen such as glycine, a methyl group such as alanine, hydroxymethyl such as serine. Classification of amino acid on the basis of properties of the group. We have neutral amino acids. They have one amino group and one carboxylic group with non-cyclic hydrocarbon chain, example glycine, alanine, and the valine. This is the alanine. And this is glycine. They have one amino group and one carboxylic acid. Can you just mute your notification sounds? With non-cyclic hydrocarbon chain. Then we have the acidic amino acid. These amino acids have one amino and two carboxylic groups. Example glutamate aspartate. Glutamate. This is the aspartic acid. And this is the glutamate. This is the then we have the alcoholic amino acids. Polar uncharged R group. These have an alcoholic or the hydroxyl group, example serine, threonine. Then we have the basic amino acids. They have two amino groups, diamino monocarboxylic and the one carboxylic group, that is histidine, arginine, and the lysine. Two amino group and one carboxylic group. This is H2N. This is structure of lysine. Then we have the heterocyclic amino acids. They have nitrogen in the ring structure, like this. That is proline. Example histidine and proline. Next, we have sulfur containing amino acids. The amino acid possess sulfur. The cysteine and the methionine. This is cysteine. Next, we have the aromatic amino acid. They possess cyclic structure with a straight side chain bearing carboxylic and the amino group. Example phenylalanine, tryptophan heterocyclic, which is the actual heterocyclic or the triosine. Phenyla. Tyrosine have OH group. Phenylalanine is like this. This is phenylalanine. A particular property of amino acid is the ionizable nature of the NH2 and the COOH group. Hence, in solution of different pH, the structure of amino acid changes. For example, in the cation, this is cation. Then it can become deuterion. It is deuterion. Lipids. Lipids are the fatty acid esters of the alcohol and related substances which are insoluble in water. Fatty acid esters of alcohol. Insoluble in water. Okay, but get dissolved in a number of non-polar organic solvents like ether, benzene, chloroform, acetone. Dissolve in the non-polar organic solvents like ether, benzene, chloroform, and acetone, etc. Okay, however, they can form colloidal complex and get dispersed uniformly in water in the form of minute droplets. Can form colloidal complex. Lipids are basically made of carbon, hydrogen, and sometimes oxygen, in which the content of oxygen is always small as compared to hydrogen and carbon. Many lipids contain small amounts of additional elements like the phosphorus, nitrogen, and sulfur. They are made up of carbon, hydrogen, and oxygen. They could be simple fatty acid. A fatty acid has a carboxyl group attached to an R group. The R group could be methyl, ethyl, or higher number of methyl groups. So this is 1 carbon to 19 carbons. For example, palmitic acid has 16 carbons. Has 16 carbons. Including carboxyl carbon. Arachidonic acid has 20 carbons atoms including the carboxyl carbon. Fatty acids could be saturated or unsaturated. Another simple lipid is glycerol, which is trihydroxypropane. Fatty acids could be saturated or unsaturated. Saturated is without double bond, and unsaturated is with one. Or other simple lipid is glycerol, which is trihydroxypropane. glycerol, which is trihydroxypropane. And the simple lipid is glycerol, which is trihydroxypropane. It is a esterified with glycerol. They can be then monoglycerides, diglycerides, and the triglycerides. These are also called fats and oils based on melting point. Oils have lower melting point, example gingerly oil, and hence remain as oil in winters. Oils have lower melting point. Example gin jelly oil. Some lipids have phosphorus and phosphorus-related organic compound in them. These are phospholipid. Phosphorus, phosphorylated organic compounds. These are phospholipids. They are found in the cell membrane. Lecithin is one example. Some tissues, especially the neural tissue, have lipids with more complex structures. Then we have nucleotides. They are the compounds of carbon, hydrogen, oxygen, nitrogen, and phosphorus. They form about 2% of the cell content. They are the units of nucleic acids. That is DNA and RNA. Nucleotides are composed of a nitrogenous base, a pentose sugar, and a phosphate. Nitrogenous base, a pentose sugar, and a phosphate. The molecule consisting of the nitrogenous base and sugar is termed as nucleoside. Nucleoside is nitrogenous base plus sugar, and nucleotides are nucleosides plus phosphate. Next, we have carbohydrates. They are the most abundant organic molecules. Abundant. They are protected during the photosynthesis of the autotrophic plants. They represent combinations of the carbon, hydrogen, and oxygen. They can be generally represented by the empirical formula CnH2ON, where N is an integer. Carbohydrates can be aldoses or ketoses. Or ketosis depending upon whether they have an aldehyde or the ketone group. In addition to aldehyde and ketone, the polyhydric alcohols, which are the sugar alcohols or polyols, in which the aldehyde or ketone group has been reduced to an alcohol group, also occur naturally in foods. Aldehyde or ketone has been reduced to alcohol. Reduced to alcohol. Based on the number of sugar units and the complexity, carbs can be classified into the mono, oligo, and the poly. Mono, oligo or the polysaccharides. Then we have the primary and the secondary metabolites. So metabolites are the intermediate products of metabolic reaction catalyzed by various enzymes that naturally occur within cells. Primary and secondary metabolites. They are the intermediate product of metabolic reaction. Catalyzed by various enzymes that naturally occur within cells. These are grouped into primary and secondary metabolites. Metabolites are converted into each other in a series of linked reactions called the metabolic pathways. These metabolic pathways are interlinked in either linear, circular, or crisscross manner. Flow of metabolites to metabolic pathway has a definite rate and direction like the automobile traffic. This metabolite flow is called the dynamic state of body consequence. This interlinked metabolic traffic is very smooth and without a single reported mishap for healthy conditions. Primary are the sum of the organic compounds like carbs, fats, proteins, nucleic acid required for basic metabolic processes and directly involved in the normal growth, development, and the reproduction. These organic compounds are called the primary. Then the secondary, for example, many plants, fungi, microbes of certain genera synthesize a number of organic compounds which are not involved in the primary metabolism, photosynthesis, respiration, protein, and lipid, and seem to have no direct function in the growth and development of organism. These compounds are called the secondary. They have no direct function. Secondary have no direct function in the growth. An example gum, resin, rubber, etc. The isoprenoids or the terpenoids are the rubber, steroids, carotenoids. The nitrogen containing compounds are the alkaloids, glycosides, glucosinolates. Then the phenolic compounds are the tannins, flavonoids, lignin, etc. We have some secondary metabolites, example pigments like the carotenoids, anthocyanins. Then the alkaloids are the morphine, sodium. Terpenoids are the monoterpenes, deuterpenes, etc. Essential oils have the lemongrass oil, etc. Toxins have the abrin, ricin, lectins have the canalavine A, drugs have the vinblastine, curcumin, polymeric substances, example rubbers, gum, cellulose. And we have the macromolecules. There is one feature common to all those compounds found in the acid soluble pool. They have molecular weights ranging from 18 to around 800 daltons. DA approximately. The acid insoluble fraction has only four types of organic compound, that is protein, nucleic acids, polysaccharides, and lipids. These classes of compounds, with the exception of the lipids, have molecular weights in the range of 10,000 daltons and above. For this very reason, biomolecules, that is, chemical compounds found in living organisms, are of two types. One, those which have molecular weights less than 1000 dalton and are usually referred to as the micromolecules or simply biomolecules, while those which are found in the acidic insoluble fraction are called the macromolecules or the biomacro. The molecules in the insoluble fraction, with the exception of lipids, are the polymeric substances. The acid insoluble fraction has only four types of organic compounds. Acid insoluble proteins, nucleic acids, polysaccharides, and lipids. The acid soluble pool represents roughly the cytoplasmic composition. Okay, the macromolecules from cytoplasm and the organelles become the acid insoluble fraction. Together, they represent the entire chemical composition of living tissue or organisms. Average composition of living cell is water, 90%, proteins 10 to 15, carbs 3%, and lipids are 2%, and ions are 1%. Then we have proteins. They are the polypeptides, they are linear chains. Holy Jesus Mary. Okay. Polypeptides, they are linear amino acids linked by peptide bond. Polypeptides linked by chains, linear chains of amino acid by peptide bonds. Each protein is a polymer of amino acid as there are 20 types of amino acid. Example alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. Is a heteropolymer, not a homopolymer. A homopolymer has only one type of monomer representing n number of times. Amino acids can be essential or non-essential. Certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. The latter are those which our body can make, while we get essential amino acids through our diet and the food. Proteins carry out many functions in living organisms. Some transport nutrients across cell membranes, some fight infectious organisms, some are hormones, and some are enzymes. Collagen is the most abundant protein in the animal world and ribulose bisphosphate. Jananda ribulose bisphosphate. Okay, carboxylase ribulose bisphosphate. Carboxylase oxygenase is the most abundant protein in the whole biosphere. Collagen is in the animal world, and which is the rubisco is in the biosphere. Protein like the collagen have functions like intercellular ground substance. Trypsin is the enzyme. Insulin is the hormone. Antibody fights infectious agents. Receptors are the central reception. GLUT4 is the enables glucose transport into cells. Polysaccharides. They are the condensation products of more than 10 monosaccharide units, such as starch and dextrin, which may be linear or branched polymers of products more than 10 monosaccharide units. They are the condensation products of more than 10 mono, such as starch and dextrin, which may be linear or branched polymers. They are also called the glycans. They are long chains which may be branched or unbranched glycans. Depending upon the composition, there are two types. It is the homo or the hetero. Homo or the homoglycans are those complex carbs which are formed by polymerization of only one type of monosaccharide monomers. Homo or the heteroglycans. Glycans. Okay. These are formed by polymerization of only one type of monosaccharide. Monomers. Example cellulose, starch, glycogen, and the chitin. And the hetero are those complex carbs which are produced by condensation of two or more types of monosaccharides or their derivatives. Example hyaluronic acid, keratin, sulfate, etc. Hyaluronic, hyaluronic, keratin, sulfate, heparin, etc. Then we have the types of polysaccharides on the basis of function. We have storage polysaccharides, then the structural, then the muco substances. Types of polysaccharides. First, we have the storage polysaccharides. Some homopolysaccharides are store form of energy. They are those polysaccharides which serves as reserve food. When needed, storage polysaccharides are hydrolyzed. When needed, they are hydrolyzed. Okay. Sugar thus released become available to the living cells for production of energy and biosynthetic activity. The two main storage polysaccharides are the starch and the glycogen. It is a storage polysaccharide of the most plant. It is polyglucan homosaccharide and is formed as an end product of photosynthesis. Starch consists of two components, that is the amylose and the amylopectin. Both of these are formed by condensation of alpha D glucose. In amylose, the successive glucose units are linked by the 1 to 4 alpha linkages, that is in the form of continuous straight chain but arranged helically. Amylopectin contains large number of glucose units in a straight chain which bear several side chains. Starch forms helical secondary structure. In fact, starch can hold I2 molecules in the helical portion. The starch I2 is blue in color. Then we have glycogen. It is polysaccharide food reserve of animals, bacteria, and fungi. It is popularly called the animal starch. Chemically, it is similar to starch. It has about 30,000 glucose residues joined together by the alpha 1 to 4 glycosidic bonds. Insulin is a fructan storage polysaccharide of roots and tubers of dahlia. Roots and tubers of dahlia and wilted plants. Insulin is not metabolized in human body and is readily filtered through the kidney. It is readily filtered through kidney.